Hello, everyone, and welcome to today's webinar titled Advances in High-Throughput Drug Screening Using TrueGel3D HTS Hydrogel 3D Cell Culture Plates. This webinar is part of the Cell Bio 2021, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by MilliporeSigma. For more information about MilliporeSigma, go to www.sigmaaldrich.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate. First, by communicating with our other attendees using our new live chat feature during the presentation. You can find this live chat located at the right-hand side of your screen. You can also participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing this presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract window below the presentation window to follow the process to obtain those credits. I now present today's speaker, Nick Asbrock, a global product manager for stem cell and 3D cell culture. For a complete biography of our speaker, please visit the Biography tab at the top left of your screen. Nick, welcome. You may now begin your presentation, sir.
Okay, thank you everyone for joining my webinar today. My name is Nick Asbrock. I'm a product manager for the stem cell and the 3D cell culture products at MilliporeSigma, which is the life science business of Merck KGaA, Darmstadt, Germany in North America. So today I'm going to talk about a new product that was recently launched called TrueGel3D HTS Hydrogel plates. This is a new product that will enable high throughput drug screening in 3D cell cultures. So an overview of 3D cell culture. So this is a recently discovered way to culture cells. It kind of bridges the gap between traditional two-dimensional cell culture models and in vivo animal models. So a lot of the advantages of 3D cell culture is that you get really just native morphology that you see in vivo animal models, but you get the added advantage of being more high throughput ready, which is what 2D cell cultures are. So a lot of people are interested in using these 3D cell culture models for a lot of drug discovery or just basic biological assays in general. So in terms of methods to grow cells in 3D, there are many different ways to grow cells in 3D. They're listed here. I'll go through each one of them briefly. So the first one is hydrogel scaffolds. These are either natural or synthetic options that will encapsulate cells. And once they're encapsulated, the cells will then grow in 3D. There's also structural scaffolds. So these are usually non-biological. These are more like structural or rigid scaffolds where the cells will be grown on the scaffolds and then they migrate throughout the scaffolds itself. You can grow cells as cell aggregates as well. So these are freely floating aggregates of cells that float around in the media. You can also grow these in bioreactors or organs on a chips. Today, I'm going to talk about the TrueGel3D Hydrogel, which is kind of the first method listed here with the star. So a brief overview of what hydrogels are. They are a form of a biological scaffold. So these are water-swollen polymers that can be derived from ECM proteins or synthetic chemicals. And as you can see on the image on the left, the cells can be encapsulated within the hydrogel itself to form this a 3D environment. And what you see when you grow cells in this 3D environment is that the cells have a more in vivo-like cellular response. And the analogy that I always think of when I think of hydrogels is like Jell-O. It's kind of like Jell-O for cells. That's kind of what it's like. Okay, so when it comes to high throughput screening using 3D models, there are some limitations on using current products or methods to culture cells in 3D using hydrogels. So they're listed here. For each model that you're trying to create, usually the hydrogel has to be optimized for all the different applications. So that would require different, hydrogel stiffnesses, hydrogel growth factors, or ECM proteins. There's a lot of upfront work that needs to be performed in order to have the right environment for the cells of your choice. Some of the materials are very temperature sensitive, so it's not the easiest to use some of these materials. Some of the materials are not transparent, they're kind of opaque, so it can be difficult to do some endpoint assays once you have the cells in the hydrogels itself, because it's difficult to image. It's also difficult to establish co-cultures. So once you have your cells embedded in the current hydrogels, it's really hard to add additional cells at a future time point to the hydrogel itself. And kind of the main hydrogels used now are also animal derived. And I know there's a trend towards more defined conditions for reproducibility concerns. Okay, so this is an overview of the new product, the TrueGel3D HTS Hydrogel plate. As you can see here, it's a 96 well black-walled glass bottom plate. So within each well of the plate, there's actually a hydrogel on the bottom of the well. And the unique thing about the hydrogel is that it has this cross-linking density gradient throughout the well itself. So on the top of the hydrogel is low density gradient, and when you go through the hydrogel towards the bottom, it gets higher and higher density. So there's this gradient throughout the hydrogel itself. And this allows you to do a couple different things. There's actually no hydrogel step required. So it's a precast hydrogel. If a customer wants to use this, it's really easy to use. You just take it out of the wrapping and take out the buffer and add cells to it. And over time, the cells will infiltrate the hydrogel because of the density gradient. It's also fully synthetic. So it's a PEG-based hydrogel. So there's no animal components. It's very consistent from lot to lot. And because of the density gradient, you can actually do sequential seeding. So it's really easy to establish co-cultures over time. It's as easy as just adding cells at different time points. And I'm going to go through each one of these in more detail in the future slides. So about the hydrogel itself. So like I said before, within each well of the 96 well plate, there's a precast hydrogel in the bottom of the well. They come delivered hydrated and ready for cell seeding. So the customer just has to remove the storage buffer and add cells and media to the hydrogel. It's as easy as that. It has this cross-linking density gradient. It is a patented technology, and this is the real advantage of the product we see. It's this density gradient. For those of you who are interested in the actual chemistry of the hydrogel, this is an overview of what the hydrogel is made of. So like I said before, it is a synthetic hydrogel. It's PEG-based, so it's very consistent from lot to lot. It is functionalized, self-functionalized, so it has RGD peptide sequences throughout the hydrogel, and it also has these degradation sites. So once cells are embedded in the matrix, they can actually degrade the hydrogel over time, and the cells can actually move throughout the hydrogel. So it is definitely functionalized for cells. Because it is synthetic, it has a very long shelf life as well, and it's very consistent from lot to lot. If you are interested in more of the chemistry behind the hydrogel and how it was developed, there is a publication here which describes the overall chemistry of the TrueGel hydrogel plates. So like I said before, the product is really easy to use, and in fact, doing the co-culture is actually really easy to use as well. So to use it, you basically remove the storage buffer, and then you just add cells and media to the wells, and you incubate overnight, and come in the next day, and the cells will infiltrate the hydrogel and find kind of its a preferred niche within the hydrogel itself. You can then come in the next day if you wanted to, and then add another cell type and incubate overnight, and you will then create this co-culture after basically 2 days of waiting. So it's really easy to use. So that was an overview of the plate itself. I'm now going to go through more of the application data for the product. So we have 7 applications listed here using the plates and cells. Today I'm going to talk about the first 4. So the first application is immuno-oncology. So we wanted to use the plates to establish a tumor cell killing assay using tumor cells and some experimental compounds. So on the top, you can see 3 conditions. The first condition is tumor cells plus the compound. In green is the cells that are dying. The middle condition was tumor cells plus non-activated T cells. The T cells are in red. And the third condition is kind of like the positive control. So this is tumor cells plus activated T cells. So the compound was thought to activate the T cells to kill the tumor cells over time. And what you see is in this third condition, you see actually increased tumor cell killing, so a higher number of green cells. And we did quantify it, and as you can see, when we spike in the compound at a higher ratio, you get a higher tumor cell killing response. So a higher green fluorescence, which means more of the tumor cells are dying. So we also did, let's see here. So we also did a kind of dose response and also different ratios of tumor cells to activated T cells. So this was using a human lung cancer cell line, the EBC1. And this just displays that, over time, you can do this really powerful co-culture method within the TrueGel plates itself. And you get really high clarity. You can get these really high resolution images, and you can see cells in real time as well. So we actually did this experiment, and we took some videos over, I think, 2 days. And this is what happens. So once again, the T cells are in red. The tumor cells are the gray ones, and the green ones are the tumor cells that are dying. So over a 2-day period, you can see how you should see more green cells pop up. So the red cells are killing the tumor cells, and they're producing this green fluorescence, which are the dying cells. And this is all within the hydrogel itself, this 3D environment. Okay, the application 2 was kind of related to application 1. We wanted to show that the plates can support just tumor spheroid growth. So we took these human pancreatic cancer cell lines, this MIA-PACA2 cells, and we cultured them over time, over 2 weeks in the hydrogel plates, and we stained for cytokeratin 19. And as you can see, the cells really do form these spheroids within the hydrogel plate itself. This is just kind of the Z-stacks of the spheroids, so top, middle, and bottom. You do see these spheroids grow throughout the hydrogel itself. So the cells will embed within the hydrogel, and then over time the spheroids will grow larger. throughout the gel. So the next experiment was taking kind of the same spheroid model, and we wanted to do a kind of a drug screening. So we took these are different cells. So these are EBC1 cells. So we seeded 5,000 cells per well in the hydrogel plates. We then did a drug screen using this investigational compound A, and we analyzed the toxicity of the cells to the drugs. So we did a dose response curve. So what you see here is with increasing concentration of compound A, you see the spheroids actually get smaller and smaller because the compound is becoming toxic to the cells. We also did a screen with 2 other compounds, investigational compounds, and you can see how they have similar effects on the spheroids. But I mean, the IC50s are different, but you do see a higher concentration of compound becomes toxic to the cells. This is just some images that you can generate using the spheroid plates. So we took that compound A and we did the dose response curve. So increasing concentrations of drugs. And this is basically what you see over time. You start seeing more of the cells dying. So they're kind of like producing these fragments of cells at these higher concentrations, which are like the dead cells. And what we actually did, we used an imaging software to actually count all these little cell aggregates. So we call it like cell bursting. This is called like a cell burst assay. So the higher number of fragments means that the compound is becoming toxic to the cells. And you can actually localize to see where the toxicity is occurring if you wanted to. And what we did is we did this analysis where we took compound A at a dose response, varying concentrations of compound A. We quantified the number of bursts at each concentration. And then we also took the spheroid size. So it's kind of, it's pretty obvious that with the more cell bursting, which is what the blue line is, at higher concentrations, this spheroid size is going down. So in theory, you could use this assay to kind of predict the most appropriate drug concentration to basically kill the majority of your tumor without becoming toxic to other cells. Okay, application 3. It's also cancer related. So there is a large interest in kind of mimicking the tumor cell microenvironment. So what we wanted to do was to do this co-culture of the tumor spheroids with the cancer-associated fibroblasts. So what we did, we labeled the fibroblast in RFP, so red, and we created these tumor spheroids. This is also the pancreatic cancer cell line again. And then we co-cultured these tumor spheroids with the red labeled fibroblasts over time for up to 2 weeks. And as you can see within the 3D hydrogel itself, the fibroblasts actually start wrapping around the tumor cells. And this is kind of thought to create that tumor cell microenvironment. And once again, because of the properties of the hydrogel, you can get these really clear high resolution images to see what's going on within the 3D environment itself. And because it's high throughput, 96 wells, you can do drug screening if you wanted to and analyze different drug responses within this environment itself. You can also analyze the extracellular matrix deposits throughout the hydrogel. So what we did here was we took some fibroblasts, which was labeled as RFP. We grew them for, I think, a couple weeks, and then you can actually analyze what ECM proteins are deposited throughout the hydrogel itself. So in B is fibronectin, in green. And C is actually collagen. C and D are collagen. D is we added ascorbic acid to the conditions to increase the collagen deposition. So when cells are grown in this 3D environment within the hydrogel itself, it definitely is producing its own ECM proteins throughout the gel itself. So really creating this 3D microenvironment. So we also have another video. So what we wanted to do was co-culture tumor spheroids with the fibroblasts. So kind of like this conditions up here to create that tumor cell microenvironment. And so for this experiment, we created the tumor spheroid. And then once it was to a certain size, we then spiked in cancer-associated fibroblasts throughout the gel itself to create this co-culture of tumor spheroids and fibroblasts. So trying to mimic what's happening in the tumor or the cancer tissue itself. And as you can see, the fibroblasts kind of surround the tumor, and they kind of like wrap around the tumor spheroid itself, and they create like this 3D microenvironment surrounding the spheroid. Let me play it again. And once again, because of the optical properties of the gel, you get these beautiful images. You can do live cell imaging with the cold cultures as well. So there's definitely crosstalk between the fibroblast and the tumor spheroids, which is what's happening in vivo. Application 4 is angiogenesis. So the plates are definitely can be used for angiogenesis assays. So what we did here was we used GFP labeled HUVEC cells, and what we did was an experiment where we spiked in different growth factors to see if it could inhibit or induce angiogenesis. So spiking in VEGF definitely increases the tube formation or the sprouting of the endothelial cells. The VEGF also increases, and then Netrin-4 is an inhibitor of angiogenesis. And we do see an inhibition over the control model. We also quantified this. It's in a paper that I mentioned before. I just didn't present the data here. Okay, so like I said before, these plates have been used in a lot of different applications, not only the ones I presented here. They've been used for numerous stem cell cultures. They've also been used for a lot of neuroscience applications as well. So creating these co-cultures of astrocytes and neurons and iPS-derived neurons. It's also been used for ADME tox applications. So we've cultured liver cells and different stellate cells, and we've done co-cultures of liver cells and stellate cells within the hydrogels itself, and it works really great. If you're interested in any of this data or other information about the product, we do have a landing page on Sigma. Here is the URL on the top, and yeah, a lot of this data is on this tech article. It's a really good reference if you're interested in the product to learn more about the features and benefits, and also the chemistry behind the plates and how to create some of these more complex 3D cell culture models that I presented today. I think now that is my last slide, so I think we're on to the questions section.